GenScript. Today, I invited Dr. Bo Wu, who is the top leading scientist at our protein production department, to talk about strategies on using chaperone co-expression to bypass the inclusion body formation during bacterial expression. Dr. Wu received his PhD in chemistry from Ohio State University, specialized in protein structure study using NMR. Dr. Wu has extensive experience in recombinant protein expression and purification in all 4 expression systems, including E. coli, P. pastoris, insect cells, and mammalian cells. In this webinar, he will share his expertise on using chaperones to boost soluble protein expression. Hope you can take this opportunity to learn from our expert and ask questions that you may have for recombinant protein expression. To ensure the sound quality, all the attendees are muted. Please feel free to type in your questions in the chat box in the lower right corner of your screen throughout the talk. Dr. Wu will address your questions altogether close to the end of the webinar. Without further ado, let's hear what Dr. Wu say about chaperone co-expression strategies for recombinant soluble protein production in E. coli. Dr. Wu, welcome.
Thank you, Lian, for your very nice introduction. It's a great opportunity to be here, and I'm glad to be here. So this is a table of content, and I will walk you through for today's webinar. We will have 6 parts. First, we will have an expression overview for E. coli, when we will use E. coli and when we should not use E. coli. And the second part will be a very important part for the E. coli expression, the inclusion bodies. We'll talk about inclusion bodies and refoldings. And the third part, we're talking about some strategies when we test expression in E. coli and do some test expression optimization. The first part and the 5th part will be the main purpose of this webinar. We are going to talk some fusion partners and chaperones, and their purposes are trying to boost the E. coli soluble expression. And 6th part, we're going to introduce some advantages of a GenScript protein service platform. So in GenScript, we have quite a different set of expression tools, and we are keeping to the trend of the most popular ones currently. We have bacterial, insect, yeast, and mammalian. We don't have a cell-free because although cell-free is a very good approach generally, it is quite costly and it's not scalable. Today, I will focus on bacterial. If you look at the PDB entries in the E. coli in all the protein structures, the E. coli expression, the recombinant protein from E. coli expression are taking majorities. That shows the importance of the E. coli expression. But sometimes we will meet a lot of different problems when we are doing E. coli expression. E. coli is really the workhorse of the recombinant protein expression, and we have a lot of different species available for you. And in GenScript, we did about 3,000 different batches of recombinant proteins, and in this webinar, we'll describe the learning from our collective experience and also our learning from literatures. There are a lot of advantages in E. coli expression. For example, the genetics of E. coli is very simple. It's very easy to manipulate, and it's very easy to scale up. You can go from probably a few milliliters to a few hundred liters. And the expression is very fast, and we will have high. E. coli is generally high yield organisms. But there are a lot of disadvantages of E. coli expression. For example, you don't have post-translational modification for E. coli. Sometimes you have to adjust the codon for E. coli expression. And for some higher organism proteins, such as the proteins from mammalian and plants, sometimes we have very low yield. Also, E. coli is great for intracellular expression. But when you want to do a segregation extraction of your recombinant protein, sometimes you have very poor extraction. There is one big problem for E. coli extraction, which is inclusion bodies. In the next part, I will give you a simple introduction about what is inclusion bodies and how we can use the advantage of inclusion bodies. So inclusion bodies is really a protective mechanism of E. coli. When the E. coli has a lot of overexpressed proteins, they will form aggregates, mostly misfolded proteins. There are some benefits of the inclusion bodies. For example, you can allow high protein concentrations, and you can protect protein from enzymatic degradation, and also you can protect cells from toxic proteins. Sometimes when the cells are expressing toxic proteins, they will force the proteins to be in the inclusion bodies. But sometimes trying to solubilize and refold the protein into the quite active form will take effort, and sometimes you have to try a lot of different ways. Why is that? Because there are a lot of different conformations that a protein chain can adopt. For example, for one angle, you know the dihedral angle, if it can take 3 different conformations for let's say the protein with 100 amino acids, the number it can take, the conformation of the number of conformation it can take will be enormous. So refolding is really a problem during the inclusion body purification. You have to try different buffers, different pH and ionic strengths. And for example, you have to try different glycerols, redox reagents such as cysteines, and different ways and different means. So we have a service that is called FloatArt. That's based on our rich experience in recombinant expression in E. coli. We can try different buffer types such as HIPS, trace, phosphate buffer, and different pH range from pH 3 to pH 10. And ionic strength sometimes important. We can try different amount of sodium chloride. And the most important thing I think is additives. For example, the glycerols sometimes can protect the protein, and the redox reagent is quite important in refolding. Also, we have different ways. For example, dilution is a very good method for refolding, and dialysis. Sometimes you have to take a chromatography to do refolding. For example, on-column refolding. We can try all the methods for you, but sometimes the result will be unpredictable, and you have to do a lot of different QCs such as SDS pH, HPLC, and the functional assay. The functional assay is very important. And all in all, well, I'll show you a case here. We did a refolding for human interleukin-5. It's a dimer in the native form. So we have tried 5 different conditions, and we found that the 5th condition is the best one. And we try to refold protein in a large scale and purify the protein by ion exchange separation, and we can get a very clean final product. And I think that's a very good case for us, but we also had some very bad experience with the refolding. So that's why we want to avoid it to some extent. So the next part will go through some the test and optimization service of our recombinant protein expression. So we have launched a package called Potential last year. So the purpose of Potential is just to try different conditions for you. So we have a standard package that we will try one condition for you probably in 3 different systems such as E. coli, insect, and mammalian. And we also have packages called Silver and Gold. In that packages, we will try a lot of different conditions for you. For example, for Silver, we will try temperature, the induction time, and the concentration of inducer. For the Gold, we'll even try different promoters, different chaperones, different strands, If look at this page, there are in the details of the potential packages. For standard, we have 4 different packages. We can try expression in E. coli, insect mammalian, and all 3 systems if we are not sure which system is the best one. For silver, like I said, we'll try the different temperature, different media, and different inducer. For gold, besides temperature, inducer, and induction time, we'll also try promoters, host cell strain, and some fusion partners, and also chaperones. So here is a case study. We did one protein for our collaborator. It's a 28 kilodalton protein. So initially we had a final yield of 1 milligram per liter, and the collaborator wants 1 gram of proteins. So if we go that way directly, we should use 1,000 liter fermentation. That's a lot. So we said we want to do some optimization. So we will try different promoters, different strengths, and also pH of the media, temperature and induction. And here are some results. So we start with 1 milligram, and we tried to optimize the promoter and induction conditions. And we can boost it to 2 milligram. And if we try for a promoter, we can boost it to 5 milligram. And then we control the growth condition. Then it increased to 6 milligram. And if we move it to fermenter, and we try the sodium density, different sodium density, and we can get the final yield of 12 milligram. So it's 12-fold boost of our initial yield. That's a lot for us. So that's just show a beauty of the optimization, and sometimes it's really necessary to save you the time and the cost. So there are a lot of different parameters in optimization. For example, the first thing we want to look at is the gene. If you get the gene from cDNA, sometimes the codon is slightly different than the codon E. coli lacks. So the first thing we want to do is do a codon optimization, and the second thing we want to look at different strands. The most popular strand should be BL21DE3, but sometimes it lacks some critical factors to make the protein working for you. And third parameter we are looking at is temperature. If the temperature is too high, the protein is very easy to go to the include bodies because you got too much proteins. If the temperature is too low, the strain is not really growing. And there are a lot of different factors such as inducers. The most popular one is IPTG. It's for T7 promoter, but T7 promoter is like a 0 to 1 change. When you add IPTG, you will have a lot of proteins, so it's a little bit difficult to control. For that, you can use Arabinase based promoter, and you have a gradient control of your expression. And the other one is growth media. We usually use a relatively rich media, but sometimes it's not good for your expression when the protein yield is very high, and you will get the inclusion body problem again. And sometimes vectors and promoters are very important, as I said before. The vectors, sometimes you want to try some high copy vectors, sometimes low copy vectors, and also the antibiotics can be a problem also. But what we are talking about today, it will be the fusion partners and chaperones. Firstly, I want to introduce some very popular fusion partners. For instance, that's all the popular fusion partners we are using right now. The first one is very popular one is his tag. So it's just 6 histidines. We put a caution sign here because sometimes the his tag can affect the function and solubility of the target protein. Although it's small, but it's a 6 histidine all along. So you have to be very careful with his tag. There are some other popular tags For example, MBP Tag is a big tag, and it can boost the solubility of your protein as I show following. And also NusA tag is a big tag. It's working similarly to MBP Tag, and I'll show later. GS tag is a good tag, but sometimes we have some problem with the stability of GS tag. It's not very stable. For flag tag, we can use anti-flag antibody resin to purify the tag, but sometimes the resin is very expensive, and the cost can be an issue. Trx tag is a very small tag, but sometimes it does not work well with large molecular weight target proteins. So let's go through tags. The first tag I want to introduce is MBP Tag. So it's an E. coli protein itself. So that means it should have a very high expression. So the molecular weight of MBP Tag is to about 50 to 60 kilodalton. So it can stabilize the fusion target protein and sometimes bind to the fusion target protein. The other similar tag compared to MBP Tag is NusA tag. So NusA is a protein which can bind to RNA. So that can, to some extent, explain its effect on on stabilizing your target protein, because it can somewhat bind to your target protein. Here shows the example when we try these two tags on the target protein. We try 5 different target protein A, B, C, D, E, and for each target protein, we saw soluble expression when we have the tag on. But when we want to remove tag, only protein C can stay in the solution. For the other 4 proteins, they will crush out from the solution. So that sort of explains that when you have MBP on it, the stability of the fusion protein increases significantly. But if you cleave them off, sometimes you cannot get stable protein, the target protein alone. The good thing is that all 5 proteins with the tag is functional in our assay. So at the end, the fusion tags indeed have our purpose to get soluble and functional proteins. The next tag is Trx tag. So if you look at the structure of Trx tag, you have beta strand in the middle, you have amphiphilic alpha helix up and down. So that explains that the stability of the Trx tag, and also it's a tag related to the cysteine. So the function of the Trx is rearranging the cysteines in the target protein. Also, you may notice that the domain of Trx presents in a lot of different chaperones. Here is an in-house example when we use the Trx tag. So this is a fusion protein, and we have a his tag in front of the Trx tag, so we can use a nickel column to purify it. And we use, I think in this case, we are using enterokinase as a protease to cleave off the Trx tag, and at the end we can get very pure protein with the help of Trx tag. By the way, without the tag, the protein is insoluble in our expression. So now we are go to the fifth part, is chaperones. So we know tags are good, and we have some problem because sometimes when we are cleaving off the tag, you are quenching out your protein out of solution. So sometimes we don't want to put the partner and your tag together. Partner and your target protein together. That's where we want to try chaperones. There are some commonly used chaperones such as PDI, GROES-L. So PDI is protein disulfide bond isomerase. It has 2 Trx domains. So in theory, it can deal with a lot of disulfide bond rich proteins. And GROES-L is a pair of chaperones. One is 60 kilodalton, the other is 10 kilodalton. The function of them is to bind to partially unfolded proteins and to stabilize them. The TF is a trigger factor. The function of the trigger factor is to bind to the nascent chain of the proteins. We don't normally use this one because sometimes the interaction between TF and the target protein is very strong, and it will disrupt the native structure of the protein. We have found some very novel chaperones such as SKP and FKPA. They are quite small chaperones, and sometimes they are very good for the secretion of the target protein. The last one I want to introduce is not really a chaperone, but less its partner, and I will touch on it later. So first, we want to look at these two proteins, SKP and FKPA. When we co-express FKPA and SKP with SAFV, the single chain fragment variable, so it's an antibody fragment. When we have no co-expression, the expression level is very low. We have these two chaperones independently with the SAFV. We saw very good soluble expression because we are using a secretive expression of the SFA. We have a signal peptide in front of SFA. The SKP or the FKPA could bind to the unfolded SFA and hybrate goes through the membrane of E. coli. So that's part of explaining how these two chaperones are working. So here there are two new chaperones. One is DSBC. DSBC is a protein, again, deal with disulfide bonds. Its function is very similar to PDI. It helps the protein rearrange the disulfide bond to adopt the native expression. And the ERVIP1P is another protein. The function of the protein is a disulfide bond oxidize. So to some extent, you can see a lot of different proteins are really disulfide bond focused. We were talking about Trx, PDI, DSBC, and the ERV1P. Based on our experience, the content of cysteine and the disulfide bonds is a very crucial factor for the solubility and also soluble expression of the recombinant protein. So make it no difference that we have a lot of experience when we have some proteins working on disulfide bonds. It will help the soluble expression of the target protein. Again, they are showing two target proteins here. One is with MBP tag in strain of BL21. We have no co-expression. The fold activity is very low. We have the co-expression of DSBC and also ERV1P. We had a very high fold activity. So this one is his tag with origami. Origami is strain which you have the reducing enzyme knockout. So we have some fold activity when you have no chaperones. But when we have DSBC and ERV1P, we have very good fold activity. That shows the beauty of the chaperones, especially chaperones which can deal with disulfide bonds. So last one is a protein called WAX. WAX. But the other name of the protein is the alanine peptidase. So it's a protein that can break the cell wall of E. coli. Sometimes it can be in good use when you have when you want to mix protein into the media. So when you have one co-expressed, you can see we have lots of protein in the media. So if you have a very high yield protein, sometimes release the protein into the media will increase yield of your target protein and makes the downstream purification easier. So right now we are in the last part. I will go through briefly the advantages of a GenScript protein service. So we have a lot of in-house technologies. For example, one of the best companies doing codon optimization, and we have different packages. For example, back power for the guarantee delivery of recombinant protein in E. coli, and fold art we just talked about that for the protein refolding, and also the system in yeast expression and bacterias. And also we have a mammalian expression system. We licensed the technology from NusA for recombinant protein and antibody production. So we have guaranteed protein package. You can get 3 milligram protein for about $2,000. If we didn't deliver the protein, there will be no charge to you. And we have large-scale protein production capability. We have bacterial fermentation up to 1,000 liters, and also yeast fermentation up to 500 liters. And we have delivered grams of proteins in 4 different systems. We also have bioprocessing services, as I talked about before. We have mammalian protein expression services for recombinant antibody production, and we also have stable cell line. The purpose is for protein production. Here showing some facilities we have. That's a big fermenter we have. That's a wheel bag we use for insect and mammalian expression. It's a bioreactor for some process development projects in mammalian system. Yeah, we just talked about that. We have 1,000 liters for bacteria, 500 for yeast, 100 liters for bacteria, and 500 liters for mammalian. So for bacteria and mammalian, we have a wheel mixture, and also different bioreactors for mammalian extraction and also process development. So here shows some details of our package. For bacteria, insect, and mammalian, we also have guarantee packages. For bacteria, it starts from $2,200. You can get 3 milligram of proteins. For the insect, you can get 1 milligram protein for about $4,000. For mammalian, we have 2 different packages. One is for recombinant protein. The other is for recombinant antibody. For antibody, you can get from up to 500 milligram recombinant antibody, and it starts from $3,000. We also have customized packages for yeast, and our PhD level scientists will review the project before it's initialized to make sure that we have successful deliveries. So this slide showing our track record for the past projects we have done. So we have deal with a lot of different projects, such as cytokines, recombinant antibodies, growth factors, kinases, and protein ligands. We have done about 5,000 batches of proteins in 4 different systems, and our success rate is overall 95%. For that, I will hand it over to Lian for the further introduction of our the services in our company.
Thank you, Dr. Wu. Here is Lian again. I'm going to spend 2 slides to overview GenScript services and upcoming seminars, as well as a promo code that you can use towards your next protein order. Meanwhile, you can type in your questions so Dr. Wu can address your questions immediately after I'm done. Besides protein services that Dr. Wu just mentioned, we also offer a broad range of services, including gene synthesis and subcloning, peptide synthesis, protein expression and purification, custom antibody production, and discovery biology services, and stable cell length, all with the goal to make research easy for you. More than 9,000 peer-reviewed journal articles have cited GenScript services and products, making GenScript the most frequently cited biology CRO in the world. I'm very proud to be part of it. Slide 37 is the last slide of this webinar. There are two upcoming webinars. One is about recombinant antibody production, presented by our senior scientist, Dr. Haixin Yu. The other one is by Dr. Rachel Spear about the expression vector, how to choose or customize vectors for protein expression. Please feel free to register if any of them is interesting to you. Also, here is the 10% off promo code that can be applied to your next order with back cover guaranteed packages, which worth between $200 to $500, depending on the package you selected. This promo will be expired at the end of day on August 15th, 2015. Now it comes to the Q&A time. Dr. Wu, would you like to take over and answer the questions raised by the attendees?
Okay. Here's a question. What are your suggestions about expression of the peptide with disulfide bonds which are necessary to be functional? That's a very good question. My suggestion is that it's very important that you try your system. If the peptide is small and it doesn't have a lot of disulfide bonds, you might want to go to a secretive production in E. coli, because the peptide should be secreted to the periplasmic space very easily, and in that space it will get oxidized. Then you have a correct disulfide bond formation. The other suggestion will be you can try some strands with chaperones ready, such as Arctic Express and the origami strain I just talked about. It's origami strain is oxidizing strain, and it can help the disulfide bond formation inside the cells. So the next question will be, what kind of translocation into peroxisome was used for the gene of interest when co-producing SKP and FKPA? Yeah, that's a very nice question. So I'm suspecting that SKPA and FKP use their own route for translocation. They should be using a different route. Normally, we are not really using the signal peptides of SKP when we're doing recombinant protein production. When you check the sequence of SKP, you couldn't find the signal peptide. So I think there should be a mechanism that SKP will segregate itself to the periplasmic expression, and in principle, it can have both ways, intracellular and into the periplasmic expression. So when you have a signal peptide such as PLB or ALE, the SKP protein can help the folds of your target protein. It can bind to your unfolded protein temporarily and help fold the protein into the periplasmic space. Okay. Yep. I think that's a question for now. And if you have any further questions, you can send me an email. That should be already shown in the beginning of the slides. And without further ado, I will hand it back to. Okay, I think there is another one. What kind of promoter did you use to co-produce SKP and FKPA? We have a lot of different choices. We can use T7 promoter, but T7 promoter is a very strong one. So if you don't want a lot of chaperones, probably you should use other promoters which you can control better. For example, you can use urbanus and maybe rhamnus, and you use a very little amount of the inducer, and then you can control the expression better. Let's see if we have another. All right. Okay, so for that, I think, yeah.
All right. Thank you so much, Dr. Wu, for your great input on recombinant protein production. I definitely learned a lot from your talk today. So I want to remind everyone that all our webinars, including both slides and recordings, will be archived on our website. So please visit our website to take advantages of these resources. Right after this webinar, there will be a quick poll on your screen. Please take a moment to complete, and we love to hear your feedback. Again, thank you all for your time and attention, and on behalf of GenScript, we wish you all success in your research.